Good evening, friends. This is Dr. Rajkumar, welcoming you on behalf of Vadodara Psychiatric Society as well as Bombay Psychiatric Society for yet another session of Brain on Wednesdays. The topic for the day is mitochondria and psychiatric illnesses. To chair the session is Dr. Kranti Kradam. You all must be knowing her. She's an additional professor in the Department of Psychiatry at St. GS Medical College and KM Hospital. She has 24 years of teaching experience, is a faculty and a PG guide at the Mumbai University, and has published a number of papers at national as well as international journals. Dr. Kranti, the platform is yours.
Thank you, Dr. Rajkumar. And it is pleasure and honor to be part of Brain on Wednesdays. So at the outset, I would like to thank IPS Vadodara as well as BPS for inviting me as an important chairperson for an important session. The topic today is mitochondria and its role in neuropsychiatric illnesses. And we have with us an eminent speaker, Dr. Malay Dave. Dr. Malay Dave doesn't, I don't have his introduction, but he doesn't need any introduction. He is the person to speak on such a difficult topic, and he's a very good teacher. All of us know, and he has the knack of simplifying this difficult subject of molecular biology, and he will definitely be giving insights onto the this subject. So over to you, Dr. Malay. Looking forward.
Uh, Thank you, Dr. Kranti. And without much ado, let's go to the presentation. Um, is it visible?
It is visible.
Yeah, okay. Once again, thank you for being here on this Wednesday evening. And we'll be talking about yet another basic aspect of neurosciences or biological sciences that might have a significant role in the pathogenesis and hence the management of psychiatric illnesses as we know. So today is the turn of the mitochondria. Why this topic? I mean, why should we be concerned about something like mitochondria? And the most important thing to understand is that any human activity, whether it is motor activity, whether it's an emotional processing or an emotional output, or a cognitive output, or a speech output, or whatever output, is a result of a number of cells, a number of systems, a number of organs working together. And when things work fine, we have a good output. Now, everything that the human body does requires energy. And one of the most important aspects of energy production, energy modulation, energy allotment is done by the mitochondria, and kind of we are forced to look at it simply because we do not have, as of now, a very coherent and a very precise pathological understanding of psychiatric disorders. Questions that are being, questions that come up these days, and what people are trying to understand is, do brain diseases have anything in common with congenital heart disease or heart disease, metabolic disorders, and other non-communicable diseases? If we look at the WHO list of disorders which have got the maximum DALYs, disably associated life years, and mortality charts and morbidity charts, in the top 10, there are going to be at least 3 or 4 psychiatric disorders, namely depression, namely psychosis, and namely dementias. Now, why do these disorders have so much of morbidity, mortality, and disability adjusted life years? We need to give it a very serious thought. And because they are on the same top 10 as heart disease, as stroke, accidents are a different thing. Serious infections, I don't think many of them are there in the top 10. So we need to have a kind of an understanding whether this kind of disorders, they group together, they cluster together. We also know from studies that the average lifespan of individuals who have got a chronic mental illness or a serious mental illness is about 15 to 20 years less than somebody who doesn't. So what happens exactly, and why does this thing happen, and whether these disorders, we name them as cardiac disorders or brain disorders, are they really distinct? Because the more we look into the basic pathophysiological processes, there are more commonalities extending right up to the genetic level. We also assume that if a system emanates from an organ, then the cause of the problem must be a defect in that organ. Meaning, we look at behaviors, we look at memory, we look at cognitive processes, we look at language, and the system that subserves these processes and the symptoms that are related to these processes is the brain. So our way of looking at psychiatric problems till now has been where these things are coming because of from the brain, and hence some disturbance in the brain might be leading to symptoms like this. The question is whether the disturbances are primarily in the brain or in something which is there in the body, and brain receives the major kind of an insult and hence produces this symptom. So this is why people are now looking at, let's say in this last 15-20 years, into basic cellular processes, basic cellular and organelle functioning, and hence the mitochondria. So this is a kind of a picture that we have studied. We have actually drawn this diagrams right from school days. And this is a nice little mitochondrion sitting in the cell and things like that. And this is basically the focus of today's discussion. So this is a double membrane bound cell organelle, means it has got two membranes, and it is seen in eukaryotes, means eukaryotes are all of us, all organisms who have got a cell wherein the nucleus has a nuclear membrane. So the nucleus is a distinct entity within the cell. Traditionally, mitochondria have been called as the powerhouse of the cell because, as I mentioned earlier, they are associated with ATP generation. The ATP, or the energy molecule, is the currency by which, due to which, all the processes in the body happen. It comes from the combination of two words, mitos meaning thread and chondrion meaning granule or grain-like. Mitochondria were first discovered in 1890 and were called bioblasts. They didn't have any name, whereas the proper term mitochondrion was given about 8 years down the line. So we have known the existence of this organism. 1890 to, let's say, 2025 will be around 200, or rather 135 years, 140 years or so. But how it works exactly and what happens is only last 15, 20, 25 years, where we have better imaging, better techniques, and better technology to understand these finer things. So mitochondria are efficient, adaptable, bioenergetic machines and have a lot of functional and compositional diversity. We'll look at that. They are of alpha proteobacterial origin and are basically endosymbiotic. Now, how did the mitochondria come into play? When there were no human beings, there were only single cells, and there were different kinds of organisms, not even dinosaurs, even before that. It is understood that there was a cell, a eukaryotic cell, and there was a bacterium, and they had a chance encounter, and the eukaryotic cell engulfed the bacterium. Then the bacterium felt comfortable because the cell was also taking it easy. Both became comfortable, and the bacterial DNA then decided to become a part of the cellular DNA. And what remained was a semblance of the bacterium, not exactly the bacterium, but bacterium in a different avatar, which was called as the mitochondrion. So basically, the mitochondria are remnants of the bacterium which invaded a eukaryotic cell billions of years ago. And since it served some purpose, this relationship has persisted. And in 1890, we understood that this thing is the mitochondrion. And the mitochondria are only one of the only two endomembrane systems. So this is one, it has got two membranes, and the other one is endoplasmic reticulum, which has got two membranes. We will look at the endoplasmic reticulum down the line, because that is also now coming up as a major player in psychiatric illnesses. And mitochondria, contrary to just 1, 2, 3 and such nice diagrams, orange colored, it's never an orange color, it's an artistic representation. But this actually form a network which spans the complete cytoplasm or the cytosol. So these are the different pictures from different sources. This is an electromicrograph. This is probably most of us have studied when we have studied anatomy and physiology. This is a more of a 3D representation. So this is the mitochondrion along with the endoplasmic reticulum. This is the network. All these green thready things are the mitochondria, so many of them. And this is the DNA. And here different cells, all these green dots, green tubular things, green filamentous things, they are all mitochondria. So mitochondria are not only that little kidney shaped diagram that we have seen, but in different formats. This is that kidney shape. This is also similar. This is different. And again, this is different. So what is the structure of the mitochondria like? Again, the similar diagram, but in a little bit of more magnification. This is how it looks. So roughly, it has an outer membrane. It has an inner membrane. It has got a space between the outer and the inner membrane. That is one space. And then there is a space within the inner membrane. So it has got two membranes and two spaces. And it has got all these green dots. It has got these brownish dots. Then it has got all these things. We'll have a look at it later on in the next slides. So it has got two distinct membranes, the outer membrane and the inner membrane, and two functionally diverse compartments, the intermembrane space, the space between the two membranes, and the space within. This is called as a matrix. So, if we, just to give you a kind of a different perspective, many of us have seen the movie Matrix and the series of films, three of them, the best ones. And the thing was that there is a matrix which kind of lays down how species and how human beings are going to be functioning. And this is the matrix actually in biological sciences, which determines a lot of what we do. So what is this outer membrane? Again, outer membrane, inner membrane, intermembrane space, and matrix. So we are talking of the outer membrane. Now this outer membrane is a smooth lipid surface. So when we look at it in the diagrams, it appears very smooth, the outer membrane. But it is made up of ancestral beta barrel protein. So this is the bacterial cell wall. It has got voltage dependent pores or channels, and very small size molecules can pass through that. Otherwise, it's a smooth lipid enriched surface. Now, along this, it's not shown here, but along this membrane, on this membrane, there are alpha helical proteins, which are more like the traditional receptors or the channels. So these are the MFF and the mitochondrial fission proteins, and mitochondrial Rho enzyme dehydrogenase proteins. Now, these proteins are important because mitochondria, like the cell, can divide. So one mitochondrium can divide into two or can fuse. So these alpha helical membrane proteins are important for maintaining the optimal function of the mitochondria, and also they can integrate functions of various mitochondria, because mitochondria can fuse. The inner membrane space. So it is the intermembrane space. So between the outer and the inner, it is a very small compartment. and it contains 5% of all the mitochondrial proteins. What also it contains and what also is important here is protein and lipid biogenesis, metal homeostasis, redox regulation, oxidative folding, apoptosis, and mitochondrial dynamics is modulated or managed by this compartment because there are so many things inside. So we have moved from here to this intermembrane space, which has got the mitochondrial proteome. And now we'll go to the inner membrane. Now, inner membrane is not smooth, it is in a convoluted form. These are called as the cristae. So the inner membrane is protein dense, it is highly impermeate. So there is nothing that goes in and out, unlike the outer membrane, which allows some proteins to go in or some small molecules to go in. And these convolutions are called as the cristae. Now the cristae have got the respiratory complexes, this button-like things, these small knobbly things, the brown ones. This is the electron transport chain, also called as a respiratory chain, which has got a number of enzyme complexes from 1 to 4 in humans, and a turbine-like ATP synthase, okay, which basically manages the energy production or ATP production. And the matrix. So attached to the cristae are the respiratory chain complexes or the respiratory enzymes, which basically lead to the formation of ATP. And then we have within that, what else? So within that, a lot of things happen. Biosynthetic reactions, mitochondrial genetics. So this purpley things, all these things are the mitochondrial DNA. We'll come to that later on. What is it and what does it do? So the main thing that happens in the matrix is the TCA cycle or the Krebs cycle. It oxidizes the carbon backbones of NADH, FADH, gradually reducing it. This forms an electrical gradient and which generates the ATP. Now, before that, what is the genetics? Now, this genetics is very interesting because as we understood before, this is an endosymbiotic process. So there are two things. One is the cell, one is the bacterium. So now when two living things are there, they are going to have two genomes. And over years, what has happened is this. That bacterium which entered the cell lost most of its ancestral genome. So it decided that I am not going to have a largely separate entity. So some of the genes were lost and most of them were lost, and many genes were transferred to the nucleus of that cell. So the mitochondria has got some DNA of its own, and a lot of it is incorporated in the main DNA of the cell. So the current eukaryotic genomes, means our genomes, have got about 600 genes of the alpha proteobacterial origin, this one. So our genes are not only ours, but they are very ancestral, coming from this protobacterium. And 99% of the mitochondrial proteins are encoded by this, which are around 1100 proteins. So again, two organisms, so two DNAs. Most of that bacterial DNA is lost. Some of it is incorporated into the cellular DNA. So these are around 600 genes. So there might have been many more which are lost, but there are 600. And this 600 code or are responsible for the mitochondrial proteins. So we saw mitochondrial proteins are many. So some of them are structural, some of them are functional, and so on. And these are about 1100. 1100 nuclear DNA coded proteins. So this is one chunk of the mitochondrial DNA. The second chunk, so this nuclear DNA, because it is important for what? It is important for the anatomical components of the mitochondria, mitochondrial metabolism, mitochondrial biogenesis, and regulation of the mitochondrion. But this mitochondrial DNA, which is kind of floating around this strands, is something unique. This is the mitochondrial DNA, and this has got critical genes for oxidative phosphorylation. Okay. And this is actually very small. This is responsible for 13 proteins compared to the nuclear DNA, which is 1100. 13 proteins. This 2 types of ribosomal RNAs, 22 types of transfer RNAs for mitochondrial protein synthesis. Now, this DNA, this one, is the key. This manages the whole of oxidative phosphorylation. So basically the energetics. So anything going wrong in a particular cell with this mitochondrial DNA has a dramatic amplification in function, right? And another important thing is this mitochondrial DNA is inherited uniparentally via the maternal lineage. The nuclear DNA will have the same kind of meiosis, gamete formation, zygote formation, and so on. So half of the nuclear DNA of each individual will be maternal and half paternal. But mitochondrial DNA is only maternal. This is very interesting because whatever happens because of mitochondrial DNA is directly inherited from mother to child, girl to the next generation. This mitochondrial gene expression, this will vary significantly in humans at both tissue and individual levels. So since this nuclear DNA is more or less uniform, these how it is going to function is more or less uniform in each and every tissue of the body. Whereas mitochondrial gene will vary at tissue level also and even at cellular levels, because this has got a wide variety. It is not this and this are not same. This is not same as this. And there are thousands of this mitochondrial DNA molecules spread throughout the cell, because the cell doesn't have only one mitochondrion. So it provides a variety of things, variety of variations. So even in the sperms, there are mitochondria. So there is paternal mitochondrion. But what happens is that this paternal mitochondria are destroyed at the time of fertilization. First at the time of maturation of the sperm and then at the time of fertilization. The human oocyte has about 200,000 copies of the maternal DNA, mitochondrial DNA, distributed after fertilization in the early embryo. So the whole lot of mitochondrial DNA, this chunk just goes. There is nothing coming from the paternal mitochondria in the initial stages, and then the replication starts. So this 20,000 will get distributed and further copies are made. And during the oocyte development, there's a mitochondrial DNA bottleneck. So what happens is when the oocyte is developing, all this thing is being distributed, but at this time there's a bottleneck. And what is the bottleneck? This bottlenecks kind of filters out pathogenic variants. So there is a system in place where in this, if there are any problematic mitochondrial DNA, they'll be filtered out. So the oocyte gets the best of the whole lot of mitochondrial DNA. And this, once we have understood, this has become a kind of a target for mitochondrial replacement therapy. So people are now looking at replacing faulty mitochondrial DNA by replacing the mitochondria, especially with respect to the mitochondrial DNA transmitted diseases. Not exactly our cup of tea, but well, there is something that is there. And what recent studies have found, this is a very recent paper, 2025, that mitochondrial DNA mutations as we become older increase in the blood and saliva, but not in the oocytes. So this is again a protective factor. One protective factor was this 200,000 copies with a bottleneck. So the oocyte, which is going to kind of fuse with the sperm, will have the best of the lot. And even with aging, the mitochondrial DNA mutations can happen in the blood and the saliva, but not so much in the oocytes. So this is again a protective factor, because the human pregnancy span, in the sense, the reproductive period is pretty long. So this kind of gives a lot of protection even at pregnancies happening at a later age. So this is one of the protective mechanisms. Another thing that we have understood is this, that the human beings, the Homo sapiens, originated in the land of Africa. And these arrows show the migratory patterns. So human beings then spread everywhere. This is the European continent, Asian continent, American continent, okay, South America, Australia. These are the years. So this is if we start at 0. Then how old is the humanity in various continents? Again, the migration is important, but what is also important is that people, when they migrated, they went to different lands, they crossed oceans, mountains, deserts, and now they are in their different environment. So people originated in Africa, they might now be in South America or Australia or the Pacific Islands or Asia. Now, these climates are differing. The environment is differing, not only in terms of barometric pressure and temperature and rainfall and altitude and things like that, but this gives human beings a different exposure to the various kinds of microorganisms also. It's not only the physical components of the environment, even the biological component. And people have been able to adapt only because of the mitochondrial DNA. This is one factor which has kind of given, again, mitochondrial DNA will be most important for oxidative phosphorylation, energy production. So whatever have been the demands on the human beings over millennia, over migration, over different kinds of exposure to different environmental challenges, they've all been able to kind of live there, not fall prey. No human populations have been extensively wiped out. Means people have been living thanks to this ability of the mitochondrial DNA to adjust by providing energy, by doing a lot of things. So in all the human beings, let's say we are here in India, and even if people have come from Africa and they are here, the mitochondrial DNA has changed. But there will be some ancestral mitochondrial DNA in all of us. The percentage is very less, but it will be there. Now, the important thing is that in those populations where the ancestral mitochondrial DNA is, the proportion is more, those were probably the populations which got infected by the COVID virus. So we heard of a lot of deaths happening in the COVID pandemic, maximum being Europeans. So here somewhere. Compared to Asia, the European continent is nearby. So the amount of ancestral mitochondrial DNA here is much more than this. And because of this, probably one of the theories is that they became more susceptible to viral pathologies like COVID. It's interesting. So what are the functions of the mitochondria? One, of course, is ATP. So these are the on the cristae. So this is the inner membrane space, external membrane, internal membrane. And on the internal membrane, we have all this respiratory complex. Now these are enzymes, which will kind of gradually reduce. So electrons get out. This forms a kind of electrical gradient. It activates the ATP synthase and produces ATP. This is the major work of the mitochondria when it comes to energy production, ATP generation. So this electrochemical production leads to the synthesis of ATP. It is also important for mitochondrial fusion and protein import. So basically management of the mitochondria or optimization of mitochondrial function. And this electrochemical potential is actually the sentinel for mitochondrial health. There are ways of measuring this electrochemical potential. Of course, it's not a test that is done on the bedside or in the clinic or in the lab, but there are ways. And if the electrochemical potential is falling, it means the mitochondria are suffering. They are becoming ill. So this is basically a chart of the mitochondrial function. Mitochondria per se, and each and every chemical reaction that happens that is important for health, maintaining health, maintaining a disease-free state happens here. So if we start from any of this side, copper and iron metabolism, reactive oxygen species. Okay, now what are these important for? Reactive oxygen species is basically signaling for stem cell differentiation. Nitric oxide for vascular tone, sulfur metabolism, amino acids for growth, signaling repair, epigenetic remodeling, and so on. NADP, we looked at energy production, so also the redox regulation. Format, so purine synthesis, calcium metabolism, heme, that is hemoglobin, peptides, lipids, and basically this mitochondrial DNA with innate immunity, because we have to remember this diagram. The older the DNA, the proportion of the older mitochondrial DNA makes us susceptible to certain things. These things are happening within the mitochondrial matrix because the enzymes are present here. But what is also important is that the mitochondrion it does have an influence and management of this folate cycle, the glutathione cycle, the methyl cycle, the purine synthesis in the cytoplasm of the cell. So many things happen within the mitochondrion. Important reactions also are guided by the mitochondria, which happen in the cytoplasm of the cell. Now, the mitochondria shape and distribution is very variable. So it is not just the kidney shape. This shape and distribution are decided on how much input is needed for various processes. So for example, pluripotent stem cells. So mitochondria are there right from day one. So when there are stem cells and these embryonic stem cells, they are going to form different kinds of other cells. Here, the mitochondria are fragmented. They are not very well developed. They are underdeveloped, but enough to provide energy for the rapidly differentiating embryonic stem cells. Even in children, there are different types of mitotypes. So in children, there are a lot of brown cells. So these chubby babies, they have got brown fats. The chubby adults do not have brown fats. They have got white fats. So this brown fat is energetically, in the sense, it is a reservoir for readily available energy. So as the children grow older, the chubbiness goes away because there are special mitotypes, different types of mitochondria, which will utilize this brown fat and manage that or metabolize that. So a lot of different functions, but mitochondria are also, they manage their own cells by fission or fusion. Now, when some part of the mitochondrion or some mitochondria become unstable, they'll need to divide so that the unhealthy part might be taken care of, removed, fission, cutting off, or fusion. So if the mitochondria feel that we need more support, two of them can fuse. So the basic activity of mitochondria is optimized by fission or fusion. Fission or fusion. So fission can happen two ways: endoplasmic reticulum mediated or this protein mediated. But we saw that on the external membrane there are two types of proteins: mitochondrial fission or fusion and the meroproteins. Now this help in fission and fusion. So basic activity of the mitochondria is modulated. If too much is happening, we do not need it, break them down. If we need more support, join them. Now this fission and fusion business is at the core of neurodegenerative disorders like Alzheimer's and Parkinson's. We'll have a look at some of the diagrams coming up. Other things, nuclear encoded mitochondrial proteins. So the nuclear DNA, mitochondrial lipid biogenesis, lipid transport, lipid contact, mitochondrial contact sites, they all help in modulating the activity of the mitochondria. So when mitochondria are no longer useful, they can be cut off. It can be piecemeal. Some part of the mitochondria can be taken over for recycling or turnover, or the lysosome can kind of eat away the whole of the mitochondria. So this is what happens at the fag end of the mitochondrial life. We looked at the immune part or neuroimmunology in one of the previous talks, and innate immunity is very important. So we saw that innate immunity is kind of there. So something is coming up, it will kind of, surveil the whole thing, see what immediately needs to be done. Then it signals the other immunity, acquired immunity, so that there's a lifelong memory form, so that further down the line, down the years, if there is a similar challenge, there is always a memory, it will take care of it immediately. But whenever there is some kind of a challenge, there's an immune challenge, there's a viral challenge, there's a tissue damage, then this mitochondria also will produce their own signals, which we have understood as damage associated damage associated molecular patterns. They have their own. Now this can trigger off an immune or an inflammation reaction. So whenever there is a tissue damage and the mitochondria are releasing their antigens, they have to be in check. This is one of the very important things, because cells are always exposed to some kind of internal or external challenges, and they are going to have an impact, especially the viral infections will have an impact on the mitochondrial integrity. So when mitochondria start getting damaged, they will start releasing antigens which can activate cellular protein recognition, or the DAMPs. And this can have a downstream cascade of innate immunity. Different kinds of things, upregulation of nuclear transcription programs, mitophagy. Okay, so mitochondria, we saw mitophagy here. The mitochondria itself can get eaten up. Okay. And release of mitokines. Mitokines are like cytokines, but mitokines are specific because they are chemical communication between different mitochondria in the same cell and different cells, so that something going wrong is communicated and they can work in tandem. Now, what happens as we become older? Mitochondria will also age and their function will get altered in a tissue specific manner. So what generally happens is, there are so many things that the mitochondria are doing. That is what we saw in this diagram, this one and this one. There is not a single reaction in a cell or a process that is important for human beings which doesn't happen in the mitochondria or in other parts of the cell without mitochondrial involvement. So mitochondria has got their hands, feet everywhere. Okay. So, and of course, they are always generating, apart from the reactions, always the ATP generation has to go on, because there are so many things happening. Now, as we become older, the mitochondria will also become old. So the wear and tear will become more frequent. Repairs will be needed more often. So the shift of the mitochondria will be more and more towards repair functions. We become older, more repair, more wear and tear. And this will happen at the cost of ATP production. This is normal aging. But one of the things that we saw in one of the earlier slides was this, that when this electrochemical potential or the energy generating potential of the mitochondria comes down, this can be a sentinel or a marker for mitochondrial health. So if this comes down and down, below a certain level, the mitochondria will start becoming ill or sick. Also, there will be changes in transcription and translation factors. And this mitochondrial aging and this mitochondrial shift towards repair will contribute to many age-related degenerative diseases. And when we are treating psychiatric illnesses or degenerative illnesses, this is going to become a problem because the chances, the rates, and the duration of side effects is going to be much more. So how are mitochondria involved in pathology? And this is where we need to really be updated, because we know that brain is an organ which takes up about 2 or 3% of the body weight. Now, that would mean that somebody who is 100 kgs will have 2 or 3 kg total brain weight. 100 kg for human being is on the side of overweight or obesity, but a 70 kg healthy adult male will have a brain weight of about 1.5 grams, 1.5 kg. Women slightly less, but about 20% of all the energy produced by the mitochondria, or 20% or 20 to 25% of all the energy requirement is by the brain. So brain being a organ which is around 2 to 3% of the body weight consumes proportionally a very large amount of energy. What it means is that even a small reduction in mitochondrial energy production, or a small problem in mitochondrial energy production and mitochondrial activity, will have a significantly larger effect on brain function. Now, this can happen anywhere. And because the energy production is falling down or the mitochondrial activity is reducing, even with slight reduction in this, we can have symptoms which are produced by brain dysfunction. Mitochondrial diseases can present at any age, in any organ system, in a highly tissue specific or multisystemic manner. So it can be one specific organ or non-specific, and it can display any inheritance pattern, Mendelian or otherwise. So whenever mitochondrial dysfunction is present, how it is going to manifest in an individual can be extremely individual. Whether it is going to pass on to the next generation, we don't have a clue. If at all, in what format, Mendelian format or non-Mendelian format, we have no idea. So what are the main things that can lead to mitochondrial dysfunction? One is, of course, genetics. So we know that there are two genetics. One is the nuclear DNA, which is more important for repair, maintenance, structure of the mitochondria, and mitochondrial DNA, which is most important for oxidative phosphorylation. So it has got two DNA. We also saw that small amount of mitochondrial DNA mutation, because it deals with only some certain 16 proteins, will have a profound impact on everything compared to nuclear DNA, which has about 1100 proteins. So one is genetic mutations. Second is aging. With aging, the focus is mainly on repair. So mitochondrial biogenesis is less. There is increased breakdown. There is reduction in fusion. With age, we saw that the number of mitochondrial mutations, mitochondrial DNA mutations, increases. In the oocytes, in the reproductive age, there is a protective factor. But as we become older, that will be lost, because this will also happen in somatic cells. And again, as we become older, we tend to accumulate. We'll keep on accumulate mutations which are happening in our chromosomes. So increase nuclear DNA, increase in reactive oxygen species. So as we become older, as lot of these processes keep on happening, we will keep on accumulating reactive oxygen species or reactive nitrogen species within the mitochondria. Now, normally, this is going to happen, but this will be cleared out by scavenging. So those glutathione and all those processes will scavenge out these free radicals till a certain point. So one is aging. Aging will have more of mutations, so mitochondria will function less. Aging is more on repair, so repair processes are in an overdrive, so more of oxygen species. And so this can trigger a lot of inflammatory changes, ongoing background inflammation with further mitochondrial dysfunction. Other things are infections, which can happen all the time, sedentary lifestyle. Sedentary lifestyle is associated with reduction in oxidative enzymes. Why? Because we are not utilizing the whole gamut of ATP. So there is a reduction in electron transport chain capacity, reduction in respiratory enzymes, metabolic reprogramming towards glycolysis, and decreased mitochondrial biogenesis. So many changes that happen with aging will also happen with sedentary lifestyle. I mean, give or take the mutations and the infections. Again, so the mitochondrial DNA has a very high mutation rate. That is one. There are multiple genes which code for each of the enzyme complexes of oxidative phosphorylation. So any change in any of these codes can have an effect on that. Apart from the brain, heart, kidney, muscle, and endocrines have high energy demand issues. These are high energy demand tissues. So brain, number one, and then these things. So mild mitochondrial dysfunction can have a problem in the brain, but it will also have a problem in heart and endocrine as the thing advances. So chronic psychiatric illnesses will also get associated as we become older, as patients become older, with heart, kidney, endocrine issues will manifest as T2DM, metabolic syndrome, even liver is important. So MASH, metabolic associated hepatic changes. The common factor being the mitochondria, how it functions. Again, the ancient, what we are still harboring from our ancestors in Africa, that mitochondrial DNA, what is the proportion, will also make us susceptible to a lot of other things. So what does happen? Mitochondria not functioning properly. We are now looking at psychiatric illnesses. Perturbed cellular function, whatever reason. This leads to neuronal energy perturbation. And what is going to happen is the networks. This is one of the earliest lectures that I had taken on connectomics. Brain networks will function properly. We assume that they were functioning properly or not properly, but they will function properly only when the energy, when the reactive oxygen species, when other mitochondrial functions are fine. To maintain a network and to keep its optimal activity will require good mitochondrial activity. If the networks get impaired, okay, then a lot of things will happen which are our higher brain functions or psychiatric functions in terms of language, memory, decision making, so executive functioning and cognition. So somebody has tried to give us how things will work. So this is mitochondrial energy, high normal, low normal, and critical energy loss. So at an operational level, cognition can be high. At a slightly regular level, healthy everyday functioning, and then gradually the cognition becomes shaky and gets compromised. But at an energy buffer, means all our functions require energy, but there's also an energy buffer. So slight reduction in energy production may not be really problematic. Below critical level, then there will be problems. So with high normal, there's an excess ATP, but at a slightly level, the energy is just enough for the functioning as well as self-restorative processes. But when the buffer falls down, when the mitochondria energy production is less, even the buffer is less, and this will make an individual susceptible to chronic stress, chronic pathogen infection, newer challenges, and degenerative disease. If it is too much of a loss, patient will be in ICU, will have a collapse. At a behavioral level, this high cognition, high focus, rumination, anxiety. At lower levels, brain fog, depression, psychosis, confusion, disorientation, delirium. So this is probably how the mitochondrial function in terms of energy, other chemical processes, reactive oxygen species, inflammation is going to affect brain function. Depression has been widely studied when it comes to the changes that are happening in the mitochondria. The most important things about depression is one, a pro-inflammatory state. Second, dampened synaptic plasticity and neuronal differentiation. And third, increased apoptosis. These three are the end results or the end processes in depression. And after these processes, we will have the symptoms. So what happens at a mitochondrial level? So many things. Impaired electron chain transfer or electron transfer chain. So decrease ATP production, increase reactive oxygen species. So there is a decreased antioxidant system, increased pro-inflammatory cytokines. There could be a damage in the mitochondrial DNA itself, and increased membrane permeability, which basically threatens the integrity of this whole system. So some of the findings related to depression. In mania, again, it is similar, but the driving forces are different. So there is overactivity of the cells. So there's an increased mitochondrial biogenesis initially, but this actually leads to a state of overactivity and increased inflammation. Cytotoxicity and apoptosis later on. So where does this cytotoxicity and apoptosis happens? With repeated episodes. So the repeated episodes in mood disorders will lay down basically a background of increased inflammation, and this will keep on driving the processes till the time the mitochondria will keep on getting damaged. Even in generalized anxiety disorders, there are findings. Substance use disorders, psychosis, autism spectrum, ADHD, developmental disorders, people have found out some mitochondrial association. Now, what happens in degenerative disorders? Alzheimer's disease is a problem wherein there is an issue with mitochondrial fission. Now, mitochondria, because of deposition of amyloid, which kind of starts a process of inflammation by nitrosylation. One of the problems that happens with inflammation is reactive oxygen species, and there is something called as reactive nitrogen species. The thing is, this is going to keep the inflammatory processes on. So there is premature or excessive breakdown of mitochondria. This will again drive the whole thing. So this keeps going on and on with degenerative changes. Parkinson's disease is similar. There are issues with fusion as well as fission happening, starting with the dopaminergic system. Again, in the liver, we talked about reactive oxygen species, inflammation, liver cells keep dying, and so with cardiac ischemia, reperfusion injury. Also in other disorders like cancers, again, not our cup of tea, mitochondrial pathology is involved. And there are a group of disorders which are strictly mitochondrial illnesses. Means there is a problem primarily with the mitochondria, and this drives the whole problem. So we have got Lee syndrome, Alpers-Huttenlocher, MELAS, mitochondrial ataxia with epilepsy, Parkinson's that we talked of, and sensory organ deficits. At different ages with different presentations. So when we are looking at this, there are these models, animal models, from which we have understood that this will be the mitochondrial pathology associated with a various with a variety of disorders, both psychiatric, neuropsychiatric, and neurodegenerative. So what people have tried to do is look at the molecular bioenergetic map. Okay, so this is how they went about doing it. They identified voxel after voxel in a 3 by 3 by 3 millimeter kind of a volume in the brain. This was the design, but what is important is this. Compared with white matter, gray matter contains 50% more mitochondria. So gray matter is biologically more active in various processes compared to the white matter. Okay. So they also studied the mitochondrial distribution and specialization, and they have a mitobrain map. This is available on the net. If anybody is interested, there is a reference which I have quoted here. You can see with images the mitochondrial activity in different parts of the brain. What are the treatment targets? First and foremost, the drugs that we use and the modalities that we use have an effect on the mitochondrial functioning. So on the electron transport chain, lithium and valproate, oxidative stress, lithium valproate and lamotrigine. On anti-apoptotic protein BCL2, lithium valproate and ECT will increase this protein. Again, this protein is a part of the mitochondrial function, and more of it means this is going to protect the cells from apoptosis or degeneration. Lithium can control hyperexcitability at the mitochondrial level. Lithium can also have an influence of calcium metabolism and BDNF, and on lactate along with valproate and quetiapine, which will decrease lactate. So lithium, 1, 2, 3, 4, almost all aspects of important mitochondrial functioning, it has got a good effect. But otherwise, antidepressants, antipsychotics to an extent, this is the indication for it, but this is probably the mitochondrial effect that happens along with these agents. Pramipexole upregulates BCL2. Then we have the nutraceutical group, so N-acetylcysteine, omega-3 fatty acids, alpha-lipoic acid, acetyl L-carnitine, adenosine, methionine. So these have been studied and found to have an antioxidant or reduction in oxidative stress kind of an effect. So many others: creatine monohydrate, leucine, isoleucine, valine, carnosine, inositol, coenzyme Q, melatonin increases BDNF, vitamin C, ketogenic diet, and so on and so forth. Now there is something called as a mitochondrial cocktail. This is used for by pediatricians for acute encephalopathy, seizures, and neurodevelopmental disorders, because again, we know that a lot of mitochondrial changes are associated with those kind of pathologies. So this is a kind of a cocktail which is used. Again, this is not a panacea, so it's not going to be useful or even helpful in many of the cases. But yes, this is a combination of things: vitamin B1, 10 milligram per kg, vitamin C, 100 milligram per kg, biotin, 0.5, vitamin E, 10, coenzyme Q, 5, and L-carnitine. Now, when we look at the kg, it's milligram per kg, and we even use it for a child, 20 kg or 25 kg, the final figure is pretty huge. It is not going to be really just 1 or 2 tablets of L-carnitine that we see available as nutraceuticals to us. So vitamin B is an important activator of pyruvate metabolism in the TCA cycle. So most important for the electron chain transfer. C, biotin, E are antioxidants. Coenzyme Q is a free radical scavenger. Carnitine binds to the long chain acyl CoAs in the cytoplasm and transports them to the mitochondria. So it will enhance mitochondrial activity. Drugs like rosiglitazone will activate neuronal PPAR gamma agonist, PPARG. Now, this is important because it increases neuronal mitochondrial biogenesis and bioenergetics. Now, rosiglitazone is an anti-diabetic agent, and in studies, it is found to be associated with a neuroprotective effect because it increases neuronal mitochondrial biogenesis and bioenergetics. And it decreases the probability that this mitochondrial function will fall below critical threshold. And that's why it is supposed to be neuroprotective in Alzheimer's. So many other compounds have been studied, okay, with regards to mitochondrial fission and fusion. Now, these two, fission and fusion, are the processes which are involved in mainly neurodegenerative disorders, Alzheimer's, Parkinson's, mainly from our perspective. Okay, so Alzheimer's, Parkinson, Huntington's, but also for a lot of other disorders, diabetes, cardiovascular disease, cancers. But people are studying this, not only the processes, but these compounds which can have an effect on mitochondrial fission and fusion. Last but not the least, the effect of exercise. A study which actually shows how over time there is fat oxidation through exercise, okay, and how lactate accumulation. Uh, so this is what happens with when people have just started exercising. Now people get tired because there is lactate accumulation. Unless fat is metabolized through exercise, there is not going to be fat loss. So blood lactate level, the more it is, and if you're able to utilize it more, then there is an exercise effect. Okay. So this was the thing, pre-exercised type 2 diabetes, beginning of an exercise program, 12 months of individualized exercise program, improved fat oxidation and significant improvement in lactate clearance. So, and there was a reversal of pre-T2 diabetes. So again, this is pre-T2DM, not established T2DM. So this will be somewhere just around HbA1c of 6. So if we are on that HbA1c of 6 or just touching or maybe a little bit more, then an exercise program over a period of time along with diet can improve pre-T2D. Why? Because one, there is an improvement in mitochondrial functioning. So this is the parameter which changes. But why it changes is because the mitochondrial function improves. Mitochondrial function becomes better if there is some challenge to the mitochondrial enzyme system, and that is in the form of exercise. So it really improves fat oxidation. It improves lactate clearance. When I am here, I'll keep on getting aches and pains, which will make me feel I should not exercise. But if I keep going on and on, even when the lactate levels are more, the mitochondria are able to process it and remove it. When I keep on exercising more and more over a period of time, it's an individualized exercise program, then the fat gets oxidized. I'll maintain a healthy weight, and there can be a reversal of pre-T2 diabetes. So that's all from my side. Thank you so much for being patient, and over to you, Dr. Kranti.
Thank you, Dr. Malay. It was an excellent and extensive overview of role of mitochondria in neuropsychiatric illnesses. You revived our childhood memories also. I still remember we used to have that question to explain the sentence that mitochondria are called the powerhouse of the cell. So it is still true. And we are exploring more and more in psychiatry regarding all the molecular biology and its role in various disorders and uh, how it can help us in treatment. So thank you very much. We'll just take if any question is there.
So even though it comes under basic sciences, even if we look in details, even if we study our psychopharmacotherapy in details, we will find that many of our molecules, as I mentioned in that chart, they have an effect on mitochondrial function. And we have been using it, whether we are thoroughly aware of the mitochondrial effects or completely ignorant about mitochondrial effects or somewhere in between. The fact still remains is that when we use the molecules, when we use the drugs, all patients do not get better. All patients do not remain better. Patients will have variable kind of side effects, and some of them will have a downhill kind of a course with a lot of comorbidity, physical comorbidity that gets associated as they become older. So yes, we know directly now, we know that yes, we are, if you want to put it in after today's topic, yes, we are able to modify mitochondrial functioning quite a bit. But
Yes.
yes, there is still a lot of scope because we are understanding, and well, maybe down the line we can have better because for mitochondrial disorders per se, they are able to kind of put in the new healthy mitochondria so that the whole functioning of the cell becomes better.
Very true. Like it is important, mitochondrium is important for all the organs, but all the more for the brain, right? Weight wise, it is less, but energy wise, it is consuming most of the energy of the body. Autism also, it helps, no?
Yeah, so yes, that's why I mentioned that
Hmm.
mitochondrial cocktail.
Hmm.
Now it becomes very difficult that when we are dealing with a developmental disorder like autism or ADHD,
ADHD.
exactly what is the kind of mitochondrial involvement? Because there are so many aspects to that. One is of course the energy production, ATP, but there are so many chemical reactions that are going on and on. So where it is going to help in which child is very tricky.
Difficult, yeah.
Because when there are mitochondrial syndromes, yes, we know that, okay, this is what is happening exactly because you identified the genes specifically and what is going wrong and what are the kind of symptoms. But when children come to us with autism or ADHD or even psychosis, then it's not very clear that what is happening. We can understand, yes, that there can be something like this behind the symptoms and producing the symptoms or contributing to the symptoms. But exactly what is going wrong? Where? Too many things. So where conditions are strictly associated with mitochondrial pathology, there is a chance that the mitochondrial cocktail will work better. What do we look at? Do we look at creatinine levels? Do we look at lactate levels? Do we look at inflammatory markers like C-reactive protein? We look at muscle breakdown in terms of creatine, CPK that we do, because these are markers of individual points or in the metabolic processes. Like CRP, HS, high sensitivity CRP is a marker for inflammation. Now, wherever that is, it can be because of any infection or
Heart.
anything. But is it specific? So do we correct it and things will improve? In some it may, in many it may not. So this kind of markers we have to study better. But yes, there are people who use this kind of cocktails. There are pediatricians I know who use these cocktails for autistic children, who will use them for ADHD. May not
Many
work in
parents
every.
ask about omega-3 fatty acids and
Yeah.
all. Can we give our kids?
Yeah. So again, when if you're looking at evidence for omega-3, it is not really useful for psychiatric illnesses. That is a current standing. Now, if you want to give it a shot, yes. But the thing is that the doses will have to be proper.
True.
And even in omega-3 fatty acid, it's not one. It's a combination of things. So we need to have the proper ratio. Then there is a chance that it may work in some properly identified kind of a case. So, this kind of things are, well, we have begun to understand. And even without understanding, we now know that, there are our drugs, so we can be happy that, we are not leaving this system untouched. But yes, we need to work on this more.
ECTs also cause changes in
Yeah, now ECT
mitochondrial level.
so in that BCL2,
Hmm.
now that is a protein. If it is more, it means the neurodegeneration is less, the cellular death, apoptosis is less. So lithium, ECTs, pramipexole, they are known to increase BCL2. So one
Okay.
of the things now is that that ECT doesn't cause any neurodegenerative change. In fact, it increases the level of BCL2. That's why some people respond to ECTs. We don't give ECT straight away, but suicidal patients, some patients who are chronic not responding, when that BCL2 increases in them, it promotes mitochondrial health. This is what we would like to think. So it's helpful in some people. One of the bases is this.
I don't think any questions are there. I think everyone has understood. We'll assume that.
We'll have a look at it again when we come to talk to other processes or
Yes.
etiology of illnesses, exactly what is happening. So this was just a kind of you can take it as a teaser trailer of. But yes, somebody can, those who are interested can read more, because all the slides have got references. So, well, they can take it up as projects just to understand, or people who are interested and have the resources to look at, uh, do basic sciences studies. In fact,
We can do the collaborative work
yeah.
also with
So,
cytology, microbiome.
for Dr. Shobha's installation, the chief guest had basically spoken on mitochondrial pathology, and she was
Yes,
from
yes.
TIFR, and what kind of work they are doing, and how they can, they're looking at different kinds of molecules that can increase mitochondrial health and so on. So, a lot of people are working on this area. Let's see what comes out of it.
Yeah, so Dr. Rajkumar had to leave, and so he asked me to do the final honors of winding up. But before that, I would just like to say, Malay, once again, a very detailed presentation. I think we had last year one on bioenergetics, which was
Yeah.
handled by Dr. Sagar.
Yeah.
So, what I just wanted to know is that when we divide each organelle and go into its dysfunction, we kind of seem to understand. Yeah, okay. Now, whatever you have told us, we have understood. I think we need to have
More comprehensive.
a
Yeah.
combined one
Combined,
to
yeah.
understand the whole thing. Like if we say that suppose there is an acute stress to the individual, when whatever happens to the individual impacts every cell.
Yeah.
And then we say what happens to the cellular membrane, what happens to the nucleus, what happens to the mitochondria.
Yeah.
I mean,
So that is the idea. So this last whatever lectures I've been taking, I'm trying to cover the main basics. So we have an idea of what are the players involved. Now, when suppose, so we looked at starting with connectomics, then epigenetics, then we looked also at neuroimmunology, we have looked at mitochondria. We'll still look at some more. But then when we touch something like stress, now once we are aware that these are the players, and we have heard about something happening, when we look at stress, so what actually happens once we know all these things, then it is just a matter of connecting of how does this, let's say, what are the neuroimmune changes, what are the endocrine changes, what is the susceptibility, and how this can manifest in different ways. This is the idea. So one is to understand all these things in parts. So actually of the whole thing, two things are very close to symptoms. One is the connectomics part, circuits, because this is the biggest thing that comes just before the symptoms. Something goes wrong, we have symptoms. We have the immune part. We'll look about the endocrine part, and then basically we look at the HPA axis later on and see how it works. So this is how we need to look at components, because one modality will not address each and everything. But if we understand the key aspects and we have something which can address that key aspect, then it can have a change in the whole, at a more grosser level.
yeah. So I think with that, we'll wind up this session. Thank you so much, Dr. Malay Dave. And thank you, Dr. Kranti, for accepting to chair the session. In fact, I have to ask at least 3-4 people every time. Most of the people are just intimidated by the topics and feel that they are not into molecular biology, so we don't want to chair the
Everybody is. If you are
session,
using
huh?
even if you're using a single molecule, we are into all these things.
That's true. That's right.
That's true. Yeah.
So we meet again on 27th of August.
No, 27th is
No,
Ganesh
no.
Chaturthi.
Ganesh Chaturthi, something Dr. Rajkumar has told you
Oh,
to
okay.
announce.
Yeah, so
Acha.
we'll take a break on that day. It's a Ganpati hai, so we
10th,
will
I think
take
10th September is the
yeah.
next
So
session.
10th September.
Okay, so meet you again on 10 September.
Thank
That's going to
you.
be taken
Thank you
by
once
you.
again.
Is the next one also by you?
It is not by me.
Okay.
We'll announce
It's a-
that.
okay.
People will get a break from me, so that's okay.
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 Thank you
Okay.
and bye bye. Good night.
Bye bye, audience. Bye bye, everyone, all who
Thank you.
joined. Thank you for
Thank you.
joining.
Yeah.
Bye. Good
Bye.
night.
Good night.